now I'm set to moderate the first technical talk session of the day. And first of all, can who's ever the Informatics Institute host, perhaps Amanda, make our presenters co-hosts so they can share their screen? So I want to start off by introducing our first speaker. Well, we have 4 talks in the next hour, 2 20-minute talks and 2 10-minute talks. As there's time at the end of the talk, we'll take questions, but timing is pretty tight. So if there's not time, we'll have to go on to the next talk. So to start off, I'm pleased to be able to introduce Dr. Stephan Barnes, a professor of pharmacology and toxicology here at UAB. Anyone who's ever attended a meeting, a workshop, a seminar, or just a casual talk at UAB always knows that Steve always has an interesting tale to tell. And he has no end of different stories that he's able to impart. And so I'm looking forward to the story that he's going to tell about acute kidney injury and the urinary metabolome of COVID-19 patients. So Steve, if you can start, my guess is you can't share yet.
No.
Can you?
No,
there you go. Okay.
I've tried, but I can't.
Okay, I'll set it this way then. All right, try now.
Okay, looks good. Maybe go there.
There you go. That's working.
Okay. Okay. Well, thank you for the opportunity to talk about what is obviously a very, very important topic in the past year, and this study has come as a result of efforts of myself, Landon Wilson from the Temple Lab, Jeff Edberg, Nate Erdman, Taylor Berryhill, and Matthew Chung. It's been supported by a grant from the Anderson Foundation, the UAB, University of California, San Diego Acute Kidney Research Center, and also by NIH for the funding. So let me go through. So most of you have seen something like this. So when a COVID patient gets sick, the virus enters the cells and it starts viral proliferation. The body responds to this. If you don't have antibodies yet, the immune system kicks in. And so you get a cytokine storm. And this may lead to you coming into hospital. And if you do, hopefully you get good project management and you'll go away and be released. But many of these patients go on to get a chemical storm, antioxidant storm created by the actions of monocytes in the blood. And this is a sort of a poisoning reaction that leads to the damage to many critical organs. And so some of these patients will recover, and some will recover but still have what they call now slow COVID. A rather large number end up dying. So this is some figures that were published recently from the Mount Sinai experience and the different hospitals that combined for this. And there were, over a period of about 3 or 4 months last year, they had almost 4,000 patients who became hospitalized with confirmed COVID-19. Net 46% of these patients were diagnosed with acute kidney injury. Of the 4,000 approximately, just under 1,000 ended up in the ICU. Of those in the ICU, there was about a 3-to-1 ratio with regard to those who had AKI versus those who did not. In terms of who died, over half of the patients who ended up in the ICU with acute kidney injury ended up dying at about a 20-fold higher risk than for those without AKI. So acute kidney injury is a really big event in this process of the COVID patient. So what we decided to do was to take a look at the metabolism changes that were going on in the patients, particularly to compare those between those COVID patients who had AKI and therefore increased risk of death, and those that did not. I think I need to define the ome here. The metabolome is much more complicated than what you've heard for the last 2 hours. The reality, the chemical nature of our bodies is of course coming in part from the human gene products that you've heard about. But the human genome actually, and most genomes of walking animals, is really quite poor. And so what we have to do is we have to eat other animals or plants. And so in our body, we will not only have human gene products, but we will have food-owned projects. But it doesn't stop there. We live our lives in conjunction with the microbiota, particularly of the lower bowel. And there is as many of those cells as there are human cells. When we go home or wherever we might be, we're exposed to the chemicals of life. And these can be personal products, such as cosmetics, or they could be things we use in our home to make our home comfortable. And then, of course, for the patients who come, the ones we're going to look at, the therapeutome. I'm sorry to add yet another ome to the, but I think it's worth saying. And the thing about the therapeutome, particularly for COVID, a lot of the time the patients who are receiving antibiotics, which of course will affect the microbiota. So how do we go about this? Well, fortunately, the UAB created an enterprise research initiative that collected urine samples during the periods that we've been dealing with the patients here. These samples were, and so we thank Nate Urban for organizing that for us. These samples were sent over to Jeff Edberg's lab. We have over 500 in the library. We went through, we were able to get information about the patients, and so they were ranked by the difference in their serum creatinine changes over a 48-hour period. And so this is a pilot study. We took 2 groups of patients. The first group were the ones that had no change in serum creatinine, and we just selected them randomly. We didn't look at the other features of these patients. And the second group were the ones with AKI, and these had serum creatinine increases greater than 0.3 milligrams over the 48-hour period, and there were 12 of those. Looking at the demographics of these patients, the upper part, the green, is the patients with AKI. There were roughly equal number of men and women. The age differences between the 2 groups was fairly small, I think about 5 years. The AKI group was slightly older than the other group. There was a marked difference in race. African Americans were 75% of the patients in the AKI group were African Americans, and white Caucasians dominated the control group. And all the deaths that occurred in these patients occurred in the AKI group. And I thank Matthew Chung for collecting this data and for working with us. So to be able to do this, this is the work that Jeff Edberg did. And they took the urines that have been obtained, put them into an epivial, added 4 volumes of ice cold methanol that allowed the virus to be inactivated and also to precipitate any proteins. This was centrifuged, the supernatant evaporated, reconstituted, and then taken over to the Temple lab where reverse phase UPLC mass spectrometry was carried out by Landon Wilson. And we collected positive and negative ion data, both collecting the molecular weight or the molecular ions, and also fragmenting those ions to get MS data. These were then matched against some accumulating public databases using the program MS-Style. And there's about, in this program, the public database has about 13,000 ions in the positive and also about 13,000 in the negative. And we also have an on-site metabolomic standards group, which allows us to add retention time into the evaluation of what each compound might be. It's very important to have good quality control for doing this. And so Taylor Berryhill was instrumental in helping me do the MS dial analysis. We used a pooled sample. So this was run at different times during the overall analysis to make sure that the mass spectrometer was collecting data in a consistent way, and it was. And we went through that data and cleaned it out a little bit to get rid of ions that were varying them to a large extent. So anything with a coefficient of variation over 10 was filtered out. And this was usually about 5% of the data. We also ran blank extractions, as if the sample was there, but we worked all the way through. We took the methanol, took some water, and pretended it was a sample. And we used that to get rid of anything that was either being introduced into the sample at any stage in the procedure. And probably that, we took out maybe about 10% of the data because of that. We all live in a contaminated world, I assure you, even in a mass spec lab. Then having got that data, we went through the data to clean out the therapeutics, the over-the-counter drugs, household and personal products as best we could. And then the data was submitted to a very nice piece of software, MetaboAnalyst 5.0, to do statistical analysis. So this is just, I'm not going to dwell on it, but these are all the drugs that we picked up in these patients that we were studying. And each person has a very different profile of drugs that they were particularly being used. The physicians, obviously, during this, particularly this earlier period of COVID treatments, they were using many, many drugs in trying to find a way to help the patient survive. When we looked at the data, this is a volcano plot. So we looked in the data in this univariate analysis. We see that there are many metabolites we were picking up which were decreased in the AKI group, and there were a few that were increased in the non-AKI group. We then went on to do multi-omics, multivariate statistical analysis, and I was really quite shocked when I did the PCA, principal components analysis. I've done several clinical trials, and it's very rare to get good separation at this early level because it's an unsupervised procedure, and of course, the patients are considerably variable. The blue dot in the center is the pooled sample, and that's where exactly it should be in this analysis. We want it to be central in the data. And that's 3 dots actually all on top of each other. Typically in metabolomics, we present the data as partially squares discriminant analysis. And when we went to that, the 2 groups completely separated, again with the pooled sample sitting in the middle of the data. So this gives us very great deal of comfort that we're measuring things reproducibly. If you then perform a VIP plot, this is a variable in protection. What it's doing, when we go back on slide, what we've done here is a lot of dimension reduction in these plots. So we have about 300 metabolites that have been identified. And of course, if we can't plot them against each other, we did that in the volcano plot. But we want to plot them in a sort of XY manner. So getting these principal components out reduces things down to an XY plot. So now we can look at the contribution of the first principal component to find out which compounds were adding the most effect for that separation that you saw. And so the ones that were increased are marked in red. And so that was niacin, amidino aspartate, pseudouridine, and several others, as you can see down there. And so niacin, of course, is a vitamin B3. The second thing is a uremic toxin, amino aspartic acid, well known. There were some bacterial components. We see indolous lactic acid, and there were others. And we also see several members of the Krebs cycle, and they were all reduced. So cisaconate, isocitrate, succinate, and citric acid. And this is if you look at the Krebs cycle as a clock, this is the first quarter hour of the Krebs cycle, a very important part. So, of course, what does this all mean? Well, niacin is very interesting because it feeds into the pathways that form NADH, and also tryptophan, if it's metabolized, which it is very extensively, can also back metabolize towards niacin. And so the fact that niacin went up in these patients says that there is a disturbance going on in the NADH pathway. And as I said, the amidino aspartate is a well-known uremic toxin, and it's a consequence of the elevated urea that these people get. It also is associated with the neurotoxicity, and this is one of the features that people report who have a more serious form of COVID. Going on, we have some other plots. This is a sparse PLSA scores plot, again showing the separations that you saw. And on the right side here, we have a heat map. So these are our pooled samples. These are the patients with the AKI, and these are the patients without AKI. And you can see very obviously, looking at these metabolites shown, that the majority of the change is going on of compounds that are going down. So these are up, these are down. And then there are 4 compounds which are showing up in this heat map, which are higher in the AKI group. That include the niacin and the amidino aspartate that I've already mentioned. Very interestingly, one of the other features of metaboanalysis is that you can use a process called random forest classification. And this is a sort of leading us into machine learning. And so what was so striking to me was that this particular group of data, when passed through this pathway, gave a perfect separation of the groups. And so suggesting that by using combination of these ions, we might be able in the future to predict somebody who is going to have AKI as a possibility. And also we hope that eventually we will be able to pick out the ones who are going to die. So summary and conclusions. Clearly the urinary metabolome is distinctly different in COVID-19 patients who develop AKI from those who don't. The prominent pathways seem to involve tryptophan and NAD metabolism, accumulation of a uremic toxin. We don't know whether this is the chicken or the egg. In other words, is it causing the renal damage, or is it a result of the renal damage? And also reduced Krebs cycle intermediates. And this has been seen before and reported in the proximal tubule of these patients. So it seems to add up. So what do we do next? Well, we've started a collaboration with Samir Parikh and Nathan Raines of Harvard. They had asked us to do some metabolomic studies on their COVID patients. And we have to say in a broad way that we got very similar results to what they did. What's most interesting is that they are looking at the role of niacin and nicotinamide as agents that can combat the effects of AKI. So it was interesting that we were able to confirm what they were already doing. We'd like to do a pilot study of how the metabolome changes with the course of the disease in the survivors versus the non-survivors. This would be very intriguing. At the moment, we're taking pictures at just a point in the course of the disease, and we didn't have full control over that because everyone was trying to sort of save the patient and not necessarily do the research at the time. And so, as I said, we're also interested to know if the AKI patients who die have a different metabolome than those that survive. We'll also pursue other hypotheses. Since many of these patients received antibiotics, we wonder what is going on in the gut microflora, and hence the production of the gut microbiome, and then the gut metabolome that gets back into the blood might be really important, and that will be a disruptor for tryptophan metabolism, which is very important to the body. And then, at the time people were doing this, they were doing this as best they could. The goal was to help the patient survive. And now we can go back and look, as we do bigger studies, about which of the therapeutic interventions were helping, and perhaps see some that were not helping. So it allows us to pursue that. So thank you. I'm happy to answer any questions. And
so I think,
yeah.
Sorry, Steve.
There's some links there for you to look.
Thank you, Steve. For the sake of time, I'm afraid we really can't entertain any questions. But thank you for a great talk. It's certainly rare, as you indicated, to see data that partitions so cleanly between different groups. And so I'm sure there's a lot more to this story to be told.
Thank you.
Okay, so next we have Dr. Darshan Chandrashekar, who's at the in the Department of Pathology here at UAB, and Darshan is going to give us an update on their UALCAN platform for working with cancer genomics data. So let's okay.
Yeah.
So you should be able to share your screen.
I hope all of you can see the screen.
Yes, we can.
Yeah. Okay. So good morning, everyone. Today I'm going to present about the recent updates that we did in our platform UALCAN. So before I proceed further, I would like to thank the organizers for giving me such a wonderful platform to present my work. So UALCAN is actually is a user-friendly web portal. It was developed in 2016 by using level 3 RNA-seq data and patients' clinical data from the Cancer Genome Atlas. So the UALCAN was originally designed to aid cancer researchers and clinicians to perform tumor subgroup-based gene analysis. I hope now you can listen to me. So to actually aid in the tumor subgroup-based gene expression and survival analysis on 20,500 protein coding genes in 33 different types of cancers. So recently, we also included mass spectrometry-based proteomics data from CPTAC for 7 different cancer types. UALCAN was actually published in 2017 in Neoplasia, and recent integration of the proteomics data was proteomics data was published in Nature Communication. So as you can all see, UALCAN is pretty extensively used by cancer researchers around the world. Till now, we have around 575,000 page views, and UALCAN has been cited in over 1,300 publications. So we have received a lot of positive feedbacks and suggestions from cancer researchers who are using the UALCAN, and these inputs actually motivated us to expand the scope of UALCAN. So what we did was to expand the scope of UALCAN to non-coding RNA. We actually downloaded the microRNA sequencing data from the TCGA, as well as extracted link RNA-based information from the 11.3 RNA-seq data. The information that we downloaded were actually processed, organized, and analyzed using open source R statistical packages and in-house Perl scripts. So we performed the gene expression and survival analysis for approximately 14,000 link RNA coding genes and approximately 1,800 pre-microRNAs. So all these analyses were actually integrated and into the UALCAN. And in addition, we also downloaded the microRNA target databases and few link RNA target databases to facilitate integrative analysis between the protein coding and non-coding genes. So all these informations now you can find, in UALCAN. So I'll briefly describe how one can access this information in the UALCAN portal. And this is a snapshot of the current UALCAN analysis page. As you can see, there are 3 distinct sections for different types of analysis. So user can perform the gene analysis, they can perform the microRNA analysis, as well as link RNA analysis. The analysis page currently has 2 panels. The panel 1 is mainly designed to provide list of overexpressed or underexpressed genes for cancer of interest. So user can click these links and they will be presented with list of top 250 overexpressed or underexpressed genes as a heat map, as you can see. So these genes are actually selected based on the difference in the median expression in normal samples and tumor samples, and actually they were sorted based on the level of statistical significance. So in addition to this, we also have option where user can extract the same type of list for specific subclasses of the cancer. As you can see in the screen, in case of bladder urothelial carcinoma, there are multiple subtypes like neuronal type, basal squamous type, luminal type. So user can click on these links and they can get separate set of top overexpressed and underexpressed link RNAs in bladder cancer. So the same applies for the other type of cancers as well. The panel 2 in the analysis page actually enables user to enter the gene symbol of the link RNAs of their interest and choose any of the 33 cancer subtypes that is available in TCGA, and they will be presented with an intermediate page where user can obtain where user can actually get the links for the analysis. So if you click on the gene expression page, so you will be presented with where you will be presented with a box plot showing the p-value as well as the level of expression of the particular gene in the normal and tumor subtypes. In addition, we also have option where user can get information for the different subtypes of the tumor groups. I apologize for the disturbance that's happening here. So user can get information about gene expression based on stage, race, based on gender, and different other subtypes of the cancer. So in addition, we also provide list of predicted targets of the link RNA. As I mentioned earlier, we have downloaded the microRNA target database as well as link RNA target databases. So for any link RNA that you query, from those databases, we have extracted the potential targets, the protein coding gene targets for the link RNA, and they will be listed. And we also provide information about their expression as per as TCGA is concerned. So not only we list the targets, but also their expression in the cancer, in the selected cancer. The survival link here in the UALCAN will provide you the Kaplan-Meier plot, where user can see the impact of the link RNA expression on the overall survival of the patients. We also provide the multivariate survival analysis results using the patient's clinical data information. And the next step here is the pan-cancer view. So UALCAN enables user to check the expression of a particular link RNA across the cancers. So that will actually aid users to select their targets. And in addition to link RNA, as I mentioned earlier, we also have information about the microRNA. So you have user can use the similar approach to query the link to the microRNAs. They can obtain the gene expression profile as well as survival profile, and we provide the predicted targets of the link RNA along with their expression as far as TCGA data is concerned.
We have 1 minute left.
So
great.
Yeah, I'm at my last slides. Yeah. So I would like to take this opportunity to all my colleagues who have helped in keeping this resource up and running, as well as my collaborators, Dr. Chad Crichton from Baylor's College of Medicine, who have been immense help in designing the tool as well as in the integration of the proteomic data, and also my mentor, Dr. Suri Varambaldi, who has been constant guide of support in all our endeavors. And thank you.
Thank you, Darshan, for updating us on your very useful and popular tool. So I think we'll now move on. And now I'd like to introduce Dr. Chindo Hicks, who's a professor at LSU Health Science Center in New Orleans. I've known Chindo for a number of years now, since the Center for Clinical and Translational Sciences has been formed. That's a partnership between UAB and a number of institutions in the Southeast, including LSU. So Chindo, please share your screen, and we'll leave it to you.
All right, so let's see. All right, I hope everyone can see the screen. I'm going to talk about harnessing biomarkers, multi-omics data for the discovery of biomarkers, and also knowledge discovery. So basically, I'm going to talk about prostate cancer and how we are integrating different types of multi-omics data. So just a little background. So prostate cancer basically is mostly a disease of men. So here, for example, is a cartoon showing where the prostate is and where the cancer is. Then we have a situation where we have the normal prostate and the a prostate that is affected by cancer. So right now, it's a major public health problem affecting about 191,000 people who are newly diagnosed with the cancer every year, and an estimated number of about 33,000 people lose their lives from this disease. So basically, our actual hypothesis basically is that we are mostly interested in looking at integrating genetic predisposition with the tumorigenesis. So essentially, we are looking at the germline mutations, and then we have the somatic mutations, and we have the epigenome. So essentially, we are saying that the prostate cancer originates from those three interrelated genomes, and the theory behind this is that if we can integrate germline mutation information with the somatic and the epigenetic information, we will then eventually be able to develop predictive models that, for example, we can integrate omics data into other risk prediction models like polygenic risk scores. So that's our eventual hope that we would like to do. But the presentation that I'm focusing on today is mostly like a hypothesis generation, where we have basically genetic predisposition happening somewhere in between here up to the time when the person actually develops the cancer. And then in between here, these factors are mediated by epigenetics. So that's the background. So obviously there are many different omics types of data, but what we are doing here is we are looking at germline mutation information again. We are then using information from the somatic mutations, and then we are combining that with methylation. The intermediate phenotype then is gene expression. So that's what we are doing here. And we are looking at both the indolent as well as the aggressive cancer. So here's an illustration of a genotype information, basically germline mutation. So this is coming from genome-wide association studies where we have case controls, and basically we find the germline mutations. We are not per se integrating only the mutations themselves. We are using the genes that contain those mutations as the unifying factor. So once we have that information, for example, in this case, for example, here you have different chromosomes and different genes, but you can also have different, you can have epigenetic alterations, somatic mutations, as well as the germline mutations. But basically the model is like this. This is the basic assumption, which is essentially this is the human genome with all the 22 chromosomes there. So different things are happening there. You have the genetic predisposition, but you also have the somatic mutation, which will happen once that person is affected by the cancer. And then before the person is affected by the cancer or before they develop the cancer, we are assuming that epigenetic factors or epigenetic alterations may alter the genome and compromise the genome integrity, and therefore triggering the somatic mutations. So this is another cartoon. This is just showing basic integration, where, for example, we have the disease gene that may be altered in the somatic genome, and then we have genetic association, essentially the SNPs or germline mutations that are associated with that disease, and then we have epigenetic information. So that's the background. So these are the data sets we are looking at. The hypothesis here is basically we hypothesize that prostate cancer originates from a complex interplay between constellations of genetic, both germline and somatic mutations, and the epigenetic alterations. The second hypothesis there is that these complex arrays of interacting genetic and epigenetic factors affect entire network states and signaling pathways, which then in turn actually drive that disease. So these are the omics types of data that we are integrating. So the design of the project itself is this way. So we have genome-wide association data here, where we are getting germline mutations. And then we have TCGA data here, where we are getting methylation data, somatic mutation, and RNA sequence data. So the first step is to make sure that the samples that we are going to be using have all these three pieces of information. And then we have this information that is coming from the outside. And then so what we have done here is basically we first compare indolent tumors with the normals. And then on the other side, we compare the aggressive tumors with the normals, and then we integrate that information. So this is just a distribution of the data set of how it looks like. So we have gene expression here, methylation, somatic mutation, and then we have the samples in indolent and also in aggressive. So in terms of the results, basically, again, this is results of RNA sequencing, where we basically just compare the normals with the indolent in indolent cancer. And then we also do the same for mutation. The idea here is to find which genes are altered in the germline, in the transcriptome, but also in the methylome. So this will be the gene. These will be the genes that are altered in both for indolent. And then here we basically repeat the same analysis, and we find the genes that are altered in the transcriptome and also in the methylome. Now, obviously, the data is multi-dimension because here we are using 450,000 methylation sites, so that's not representative of the entire methylome. So after we have done that, then we are going to take these genes that overlap between this. So we found these genes that contain both, and these genes here, and then basically we create here to see which genes they overlap between indolent and the aggressive cancer. So then after that, we go to here to make the assessment of what is the impact of DNA methylation on gene expression. So this is basically for the indolent, and we are looking at the different impacts here. And this is for the aggressive. Again, we are also looking at the impacts of DNA methylation on gene expression. So once we have determined that, then we ask the next question, which is, so which of these genes that are methylated are affected by DNA methylation are also impacted by somatic mutations. And this is basically in this cartoon, as you can see here, now we add the somatic mutation information, RNA-seq data, and methylation data in indolent, and we do the same here for aggressive and somatic mutations. Here, basically in this cartoon, we're simply looking at which genes overlap the two, but which genes have all the three pieces of information are unique to each subtype of cancer. For example, in this case, 845 genes are unique to indolent, and here 1,284 are unique to aggressive, and then we have these genes that are overlapping. So we then basically, fourth level integration, we look at what about if now we add germline mutation information? So here you can see three-way interactions of germline, somatic, RNA-seq, and methylation, and indolent, and the same thing in aggressive. Again, we try to look at which genes are unique to each type of prostate cancer. Once we have done that, we can obviously map their positions to see, because we know where the methylation sites are and where the expression values are, but we also know where the mutations are. So this is just a cartoon showing that in one chromosome, showing where the positions of those mutations are matched with the gene expression and the DNA methylation. So after we have done that, we ask the question, say, do these genes actually interact in the gene regulatory networks or in the pathways? So this is basically a pathway input or a network input where we simply input the genes that have all the somatic mutations, germline mutations, the epigenetic alterations that are also associated with a particular cancer, in this case associated with indolent cancer. And then we find the those networks. And then here is the same thing that is done separately. Now here in both cancers, we are only using those genes that are unique to each subtype of prostate cancer. So that's how we are approaching this. And then of course, in conclusion here, we have shown that the integrative analysis is a powerful approach for mapping possible oncogenic interactions between genetic and epigenetic factors in those subtypes of cancer. We also look at is that prostate cancer is an emergent property of molecular networks and signaling pathways that are under genetic and epigenetic control. We haven't mapped the physical interactions yet, but we do see that genes that contain those alterations interact. So we basically conclude that the harnessing high-dimensional multi-omics datasets has the promise of linking genetic susceptibility to tumorigenesis. Further studies, as I mentioned, will be to how can we integrate this information now to development of prediction models such as polygenic risk scores and those other models. What's going on here? We'll talk about this in the next session. So we have a master's program that we have developed here for those students who are interested in this. And this is the team that is working on this. Somewhere here in the hidden corner here, there's a gentleman whom you probably have seen here called Tarun Mamid. He did this work. Most of this work was done by him when he was here with us, but now he's a PhD student at UAB. So this work now is continuing to where we are looking at mutation-based as well as mutation-based and epigenetic-based classification of indolent versus aggressive cancer. So obviously we would like to acknowledge the funding. We do have support from the CCTS at UAB and then the various funding entities, the School of Medicine, the Lefkowitz organization, and then the National Cancer Institute, a national foundation that we just got a grant recently from the National Science Foundation. So, I would like to stop there, and if there are any questions, then that's it. But if not, then we will discuss that later. Thank you very much.
Thank you, Chindo, for really providing us with a great example of integrating data from multi-omic sources, something that's certainly now critical to biomedical research. I'm going to suggest if people have questions for Chindo to enter them into the chat. And then they'll be able to sort of type out their answers. But for the sake of keeping everything on time, I think we'll move on. So thank you, Chindo. So next we have up Dr. Zach Wallen, who is now a postdoc in the lab of Dr. Hadepami in the Department of Neurology at UAB. Zach was not too long ago a graduate student in Hade's lab. But he has since graduated and is now moving on. But he's going to talk about some of his work looking at the methods for statistical analysis of microbiome data sets derived from Parkinson's disease patients. So Zach will let you continue. It looks like I know long. Zach, can you try to share your screen now?
Okay, there we go. I was waiting for the other shared screen to.
Yeah.
Let's see. So I should have it now.
Great.
I'm hoping you see my slides.
You do?
All right. Yeah. So this research I'm going over today was part of my dissertation when I was a graduate student. I'm really only getting to go over just a portion of it for sake of time. But yes, this is a comparison study that I performed looking at different differential abundance testing methods using two large Parkinson's disease microbiome data sets that we have at our disposal at Pathology Lab, which have been derived from 16S sequencing. And again, I can only go over a portion of this due to time, but it is undergoing revision for BMC Bioinformatics. So hopefully it'll be published there soon and you can check out the rest of it. So anybody who does microbiome research pretty much knows what differential abundance testing is. It's basically a cornerstone analysis that's performed in any microbiome study where you're looking at individual taxonomic differences between two or more groups, no matter if those two groups is a disease versus individuals who do not have a disease, or if it's maybe a certain tissue. But basically, it's the goal of trying to determine what individual microorganisms, or maybe even clusters of microorganisms, are different or differentially abundant between two or more groups. And just have a little schematic here that kind of represents that. You have ecosystem A here that have these little green microorganisms here, compared to ecosystem B, who have these reduced. So that's what I'm referring to when I'm referring to differentially abundant microorganisms and testing. So how does one test for differential abundance? Again, if you've heard a microbiome study, you've already know this, but just in case there are individuals who are not familiar with this, you basically just perform serial univariate statistical tests for each detected taxon that you are interested in a particular group of samples, which could range from hundreds to thousands, depending on your method of detection, your sequencing level, whether or not you're looking at higher taxonomic levels like phylum or kingdom, or lower versus genus or species. And basically, you're testing if the relative abundances of these taxa are significantly different between your groups that you're interested in. And then usually you correct for multiple testing, which is usually a conversion in FDR Q value or a false discovery rate Q value, because you've just done thousands of tests, and without that correction, just by chance, you're going to find something that was been detected. So a problem comes in is that there's loads of these tests or methods out there to perform these types of analysis, and they just keep growing every day because microbiome is a very hot field right now, and there's a lot of method development going on. So a problem is, which one do you choose, or which ones do you choose to perform these types of tests? And this is a problem that we ran into in our lab because there was just so many and we didn't really know which one we should choose. So this is really, this whole project was basically derived off of self-interest for the most part. But yes, choice of differential abundance testing methods can influence results. So depending on which method you choose, you might get different results. And this can cause an inter-study variation in results, even when the study is on the same conditions. So these methods can range from your standard statistical tests that you probably all heard about, negative binomial generalized linear models with or without accounting for zero inflation, just regular GLMs, just t-tests, Kruskal-Wallis or Wilcoxon rank sum. And then these tests, usually there's some type of normalization or transformation applied, three of which I'll focus on here is total sum scaling, or basically turning counts of microorganisms into proportions, the centered log ratio transformation, and a more newly described robust centered log ratio transformation. You also have RNA-seq methods that have now been repurposed for microbiome studies, since the underlying data is fairly similar. Either you're looking at counts of microorganisms or counts of gene transcripts. So these methods have been repurposed for microbiome studies. And microbiome-specific methods, which have been created for the study of microbiome. And you have really one method at the moment that's been touted as one that's for both RNA-seq and microbiome, which basically it's just looking at compositional data in general, which both RNA-seq and microbiome are. And all of these methods, this is not an exclusive or completely inclusive list. There's many more methods out there. These are just some that I've included into this comparison study because they seem to be the more popular ones that you see in the literature. So basically, what I was interested in was seeing what's the concordance between these methods. How do they hit the same targets? Do they detect the same things? Are they vastly different? And it's important to understand how these different methods compare to one another, so you know what's the likelihood that their answers are going to line up for what they're detecting as significantly different. And comparison studies have been performed previously looking at a handful of methods here and there, but it's really only a few at a time, and it's usually on simulated data. So there wasn't really before this any that were done on real data sets, especially ones collected to study a complex disease like Parkinson's disease. So the goal of the study was to do that, to perform a fair bit of these methods found in the literature on a complex disease data sets and compare how their results pan out to each other. So what I did was gather commonly used differential abundance testing methods from the literature and use them to test for differentially abundant genera. So if you're not familiar with taxonomy, genera is one step above species between PD patients and healthy controls and 2 large Parkinson's disease gut microbiome data sets, which is about 300 and some for one data set samples and about 500 samples for the other data set. Once those differential abundance testing methods were performed, I calculated concordance between those for each data set and across data sets. So first, I performed them on unfiltered data, so it's about around 500 genera in each data set, 300 to 80 in common. And this is basically the summary of that. So the top is the pairwise concordance for each method for data set 1, the middle for data set 2, and the last one is for data set 1 and 2, taking replication of detected signatures into account. And I have them ordered from left to right on higher mean concordance, which concordance here is on the left and is actually referring to Pearson correlation coefficients. So we have some methods that consistently had above average pairwise concordance with other methods, which formed a lot of the non-parametric methods, and also some of the standard statistical methods like GLM and t-test and Kruskal-Wallis. They use the total sum scaling and centered log ratio transformations. We had also some that were consistently below average in concordances, which was a fair number of a few of the RNA-seq methods, all of the standard statistical methods that use a robust CLR transformation, and then one method that's a zero inflated Gaussian method that's called FitZig, as well as the negative binomial generalized linear model with zero inflation. So next with that, after that, I did the same analyses, but on a filtered data set just to see, would we get the same results if the taxonomic data had been filtered before, so removing out more of the rarer taxa. So with that, there was about 130 genera in data set 1, about 200 in data set 2, and 130 in common between the two. And then the same concordances were calculated in the same manner. And again, we had a fair bit of the ones who were above average in the last instance still there. However, DEC2, ANCOM, and then a newer ANCOM BC, which stands for Analysis Composition of Microbiomes, kind of dropped out a little bit from that running, showing that the 10% filtering had an effect on them. And then the below average still held some members of the below average as the last scenario, but now that ANCOM has been thrown into the mix, showing that it was severely affected by the filtering of the taxonomic data beforehand. Like I said, there's more to this, but unfortunately I do not have time to go into that right now, so I'll just finish up that top there, and then just my acknowledgements, the NGRC consortium, which is where this data has been derived from, funding, Payami Lab past and present, my committee who helped along with this chapter. And then eventually, once this is published, the source code for all of these analysis will be deposited in a GitHub repository that can be accessed publicly. Thank you.
Thank you, Zach. I hope your paper gets published pretty quickly, so when we have a reviewer complaining about our papers, we can now reference your paper.
That's what I'm hoping it can help out with. So
there we go. So thanks to all the presenters over the last hour. And now my role is to pass the baton to Dr. Millet Basu and let him moderate the next set of talks.